viewers, welcome to Medikine 24/7. Today we will be discussing the topic shock. Please watch the video till the end and follow us on Instagram at Medikine 24/7 for more exciting medical contents.
Hello guys, welcome back to part 2, and in this video we'll focus on the chemical and molecular alterations in shock and some common types of shock. Let's start with cellular and molecular alterations that are seen in shock. In our previous video we discussed about the gross pathogenetic mechanism of shock, and in this video we'll talk about the cellular and molecular pathogenesis of shock. There are 4 primary cellular and molecular alterations that are seen in shock. These 4 are alteration in cellular structure, alteration in cellular metabolism, humoral vesogenic factors, and inflammatory response. Alteration in cellular structure includes loss of cell membrane integrity, mitochondrial damage, and lysosomal injury. Alterations in cellular metabolism includes deficiency of oxygen and ATP supply and reactive oxygen species formation. The vesogenic factors that contributes to the pathogenesis of shock includes several vasoconstrictive and vasodilatory factors, and inflammation mainly consists of systemic inflammatory response syndrome, or SIRS. It's a widespread activation of inflammatory responses or reactions in the body. If the shock is not compensated, then eventually all alterations in the cellular membrane leads to increased disbalances in the osmotic balance and subsequent rupture of the cell. And rupture of the cell leads to exposure of the intracellular contents, especially lysosomal contents, into the bloodstream, causing activation of mast cells and subsequent release of histamine. It also causes widespread activation of inflammation in the body, which eventually leads to SIRS, or systemic inflammatory response syndrome. Hence, cell death primarily occurs via 2 ways. The first is apoptosis and the second is necrosis. Apoptosis is primarily mitochondria mediated due to increased oxidative stress, as if there is decreased tissue perfusion and decreased oxygen supply to the cells, there is increased production of reactive oxygen species by mitochondria, and also the actions of lysosomal enzymes on nucleus leads to DNA damage, and by the action of p53 gene, apoptosis cascade is initiated. Necrosis is primarily mediated by cellular rupture due to increased cellular swelling and loss of membrane function, as we said before. The primary difference between apoptosis and necrosis is necrosis leads to initiation of widespread inflammation in the body. As we said before, that rupture of the cell leads to exposure of intracellular lysosomal contents, which leads to activation of the inflammatory cascades in the systemic circulation. But in case of apoptosis, there is no inflammation. It's an intracellular process, and the cell dies. After the death of the cell, there are neutrophils and macrophages which clears the debris of the apoptotic cells. In this slide, under a scanning electron microscope, we can see an injured cell and the mitochondrial injury that the cell has sustained. We can see evidences of mitochondrial swelling. You can clearly understand that the mitochondria are bigger than usual, and also we can see evidences of mitochondrial rupture, like here, and subsequent release of intermitochondrial contents into the cytoplasm. There are 3 primary factors which are responsible for altered cellular metabolism, which are mitochondrial dysfunction, decreased ADP formation, and oxygen deficiency. These are the 3 primary causal factors which leads to altered cellular metabolism. The primary metabolic change that occurs in case of decreased oxygen supply and decreased ATP formation is uncoupling of aerobic respiration and increased anaerobic respiration and subsequent lactic acidosis. How increased anaerobic respiration and uncoupling of aerobic respiration leads to lactic acidosis, let us look at that mechanism briefly. Whenever there is decreased supply of oxygen, there is increased amount of glycolysis because Krebs cycle cannot occur without oxygen. So glycolysis takes over for the primary source of oxygen. Whenever there is increased glycolysis, there is increased amount of pyruvate formation because the end product of glycolysis, as we all know, is pyruvate, and as Krebs cycle does not occur, pyruvate starts to accumulate. And pyruvate eventually converts to lactate by the process of lactic acid synthesis, and the lactic acid starts to accumulate in blood, and which is a very early sign of hypoxia. Now let's look at the Cori cycle briefly, because Cori cycle helps us to summarize the glucose to lactate and lactate to glucose turnover in the body in normal conditions. In normal condition, whenever there is increased muscular activity, the muscle cells uncouples the Krebs cycle and mainly focuses on glycolysis for rapid and increased amount of ATP supply temporarily. And as a direct consequence, there is increased accumulation of pyruvate, which eventually leads to lactate formation by lactic acidogenesis, and the lactate goes out to blood and eventually to liver. Well, in normal condition, the small amount of lactate which is formed by the skeletal muscles in liver is converted back to glucose by the process of gluconeogenesis. In this method, in this way, it consumes 6 ATP. But in gluconeogenesis, the glucose formed in the liver goes back to muscle again and undergoes the cycle all over again. Well, this is the normal condition that is seen in the body whenever there is increased oxygen need of the skeletal muscles, and in order to boost the temporary ATP supply, the Cori cycle takes over. But in case of shock, the accumulated lactate is greater than 5 millimole per liter, which is too much for the liver to metabolize and convert back to glucose. So it starts to accumulate in the blood, and eventually it leads to lactic acidosis. Whenever there is too much lactate in blood, that is greater than 5 millimole per liter, it is too much for the liver to metabolize, and the Cori cycle is disrupted. Reactive oxygen species are produced in primarily 3 ways, that are ER stress, release of mitochondrial free radicals, and activation of several enzymes. ER stress is caused whenever there is increased amount of misfolded proteins compared to the amount of folded proteins. So in order to fold the proteins in their proper conformations, the endoplasmic reticulum needs constant supply of ATP and oxygen. But whenever there is absence of oxygen or decreased ATP formation, there is increased amount of misfolded protein in the ER that starts to accumulate and leads to ER stress and reactive oxygen species formation. Due to mitochondrial damage by calcium deposition or increased osmotic disruptions, the free radicals that are present in the mitochondria starts to enter the cytoplasm, which also leads to increased amount of reactive oxygen species in cytoplasm. There are several other enzymes that are activated, which are xanthan oxidase, INOS, or inducible nitric oxide synthase, and NADPH oxidase. All of these enzymes leads to formation of reactive oxygen species. Oxidative state of the cell is primarily reflected by the glutathione, by the oxidative glutathione and reduced glutathione ratio. Glutathione is an antioxidant that is present in a cell, and reduced glutathione reacts with reactive oxygen species, for example hydrogen peroxide, and it is converted to oxidized glutathione. Oxidized glutathione is converted back to reduced glutathione by the conversion of NADP plus to NADPH. This is a very important antioxidative mechanism that helps to eliminate the reactive oxygen species in the cell. But whenever there is increased amount of free radical production, the antioxidants starts to run out quickly. So there is increased accumulation of free radicals or reactive oxygen species, and there is progressive decrease in the amounts of antioxidants. This condition, that is decreased antioxidants and increased free radical production, leads to cellular oxidative stress, and the ratio of oxidized glutathione and reduced glutathione increases in case of oxidative stress. But whenever the situation starts to normalize, the ratio of GSSG and GSH starts to decrease. Now let's look at the several humoral vesogenic factors that contributes to the pathogenesis of shock, which are several vasoconstrictors and vasodilators released by different cells, and these vasoconstrictors and dilators controls the vasogenic activity of blood vessels. The vasoconstrictors include catecholamines or epinephrine and norepinephrine, endothelin, and angiotensin II. The vasodilators include nitric oxide, histamine, 5-HT or 5-hydroxytryptamine, which is also known as serotonin, and beta endorphins. The function of the humoral vasogenic factors and vasoconstrictors and vasodilators acting on blood vessels and leading to widespread alterations of vasogenic activity, causing microcirculatory disturbances. We have discussed all these mechanisms in grave detail in our previous video in the shock part 1. So please check out that video in order to get the detailing of the activity of the humoral vasogenic factors that primarily acts in shock and how they causes the microcirculatory disturbances. The inflammatory response starts from cellular hypoxia, which causes release of several chemoattractant cytokines, which leads to recruitment of macrophage and neutrophils. The enzymes released by neutrophils after recruitment leads to cellular membrane damage, and whenever there is increased cellular membrane damage, there will be increased reactive oxygen species production. Now let's discuss some common types of shock and their clinical features. The first is hypovolemic shock. Well, it is a typical pathogenesis and manifestation of shock seen in a wide variety of shock. It contributes about 30 to 40% of the shock that are seen in hospitals. The initial change is reduction in blood volume and subsequent reduction in blood pressure. It tends to get complicated by acute renal failure and endotoxemia. Acute renal failure, whenever there is decreased blood volume flowing through the kidney, or whenever there is decreased renal vascular pressure, there is damage to the glomerular filters, and the increased damage to the glomerular filters leads to acute renal failure. Endotoxemia, or presence of endotoxins in blood, leads to further widespread activation of inflammation and further reduction of blood pressure or vasodilation or widespread vasodilation. So these two are the most primary complications of hypovolemic shock. And hypovolemic shock is primarily caused by burns, widespread burns, postpartum hemorrhage or any kind of hemorrhage that leads to increased amount of blood loss, and vomiting and diarrhea. Whenever there is increased amount of fluid loss, as a direct consequence, there will be decreased amount of blood volume or hypovolemia. And after a certain extent, the hypovolemia leads to hypovolemic shock. Another common type of shock that is very commonly seen is septic shock. Septic shock is caused by bacteria or presence of bacteria or bacterial antigens in the systemic circulation, and it involves several inflammatory and counter-inflammatory responses. It is primarily caused by gram-negative bacteria and widespread septicemia caused by gram-negative bacteria. There is a component that is present in gram-negative bacterial cell wall, that is LPS or lipopolysaccharide. It's also known as endotoxin. Whenever there is increased amount of endotoxin present in blood or endotoxemia, there is several inflammatory and counter-inflammatory responses towards it, which leads to widespread vasodilation, that is increased vascular capacity, and decreased blood volume and cardiac output. The septic shock often tends to get complicated by SIRS or Systemic Inflammatory Response Syndrome. The third common type of shock is anaphylactic shock, which is brought about by anaphylaxis or widespread allergic reaction. In case of anaphylaxis, the mast cells are widely activated by immunoglobulin E or IgE, which leads to release of histamine. As we mentioned before, that histamine is a vasodilator, which leads to vasodilation, increased vascular capacity, increased capillary permeability, which leads to swelling and edema in different parts of the body, which further leads to decreased amount of blood volume. So vasodilation and decreased blood volume expedites the decrease in BP or blood pressure. Blood pressure in this case decreases quickly and rapidly because vessels starts to dilate from the beginning, and the edema expedites the decrease in blood pressure. The fourth common type of shock is cardiogenic shock, and in this case cardiac pumping function is decreased or cardiac functionality is hampered, which leads to decreased cardiac output. Whenever there is decreased cardiac output, there is decreased perfusion of the tissues, and there is decreased oxygen supply in the vital tissue beds like brain or kidney or the GIT vasculature. Whenever there is decreased cardiac output, the blood pressure decreases significantly in the early stages, and it tends to be complicated by congestion of lungs and other organs. Because the heart isn't functioning properly, there is congestion in the venous capacitance system, which leads to congestion of the liver, spleen, and lungs primarily. Neurogenic shock is primarily caused by spinal cord trauma or trauma to the central nervous system, gunshot wounds to the spinal cord, sports injury, and several medications to the autonomic nervous system that helps to regulate the breathing. The initial changes are in the vasomotor center, which leads to widespread vasodilation and increased vascular capacity. It often gets complicated by fainting or coma. Thank you for watching. Don't forget to watch our part 1 of shock, where we have discussed the etiology, causes, and classification of different types of shock in detail, and we have also discussed the pathogenetic mechanisms of the 3 primary stages of shock. If you guys want the PPT of the topics we discuss on our channel or notes of the corresponding topics for free, you only need to subscribe to our YouTube channel and follow on Instagram at Medikinate247 and DM us the topic. If you found our video informative and helpful, please like, share and subscribe to our channel for future medical video lectures. Don't forget to press that bell icon. See you on our next video on Medicaine 24/7. Stay healthy, stay safe.